have you in this platform.
Thank you.
In 58 Navodaya Vidyalayas all over the country, Vigyan Jyoti project launched by Prime Minister of India has launched since last year. And this is exclusively for the girls' children studying in +1 and +2, madam.
Okay.
So, and every week we are arranging such a prestigious talk from the eminent personalities like you, madam. It's an honor for us that you agreed to consent to address our children. So, Navodaya Vidyalaya, I hope you may be having some idea about Navodaya Vidyalaya.
Yes.
Apoptosis organization working under HRD. So this is a prestigious organization and doing very well in the senior secondary school level. For your kind information, madam, this Navodaya Vidyalaya recently published CBSE class 10th and 12th results. In class 12th, we are the #1 in the country with an average of around, school average of around 80% subject average. Almost. And even yesterday, Prime Minister talked with one of our boy, a topper of class 12th commerce stream. So we heard about our organization. This program is exclusively for motivating the children to the basic science through this Vigyan Jyoti project, so that people like you coming forward, a personality reach the highest top in our scientific community, is the best person, no other better person than you for our children to live. Dear children, I would like to introduce about Madam Chandrima Shaha, a professor of eminence. She was the former director of National Institute of Immunology, having a very vast experience in the science, in the field of immunology, parasitic, especially Leishmania species varieties for disease control, and find out, especially in the cell biology area. Recently, Madam has been selected as the president of Indian National Science Academy, a very, very prestigious position held by male members for the last 5 years. We are very proud to welcome you, madam, to see that you are on the top of the scientific community, especially doing the. And dear children, madam, publications are more than around 80 to 100 publications at national and international level. A well-acclaimed professor at the international level. Even after getting know that you are going to talk today, gone through your profile, I was very astonished to see your profile and your achievements, madam. And definitely our girl children, only the members of this group is only girl children who are the members of the Vigyan Jyoti project. We want these children to come up in their life to take up this as a career in the future and get admission in prestigious institutions. So I would like to enlighten our children in that area also in your talk. So now Navodaya is conducting a Science Congress program also for the best 100 a 5-day program in prestigious institution like Indian Institute of Science, last year Indian Institute of Science for 5 days, an in-house program for 5 days for this to acquaint well with the scientists, know the scientists. We only heard about scientists and visited many institutions. So it's we are very happy to welcome you to address our children. It is an honor for us you are in our platform. On behalf of Navodaya Vidyalaya Samiti, I welcome very sincerely to this program, and our say Ramchandra Sahai Commissioner is also this platform. Request Ramchandra Sahai to have Madam take over the stage.
Sir, may I request our ex-joint commissioner, sir, Ramachandra, please, sir, a word, please.
It's privilege to welcome ma'am. Hope Dr. Chandrima ji rather I remember I was in INSTEM for some time. So I used to coordinate some academic advisory committee and executive council meeting from there. She joined there also. So ma'am, I worked here as a joint commissioner, and I retired and now somehow continued here And I'm also associated with this project of Vigyan Jyoti, where students are encouraged to choose science as a career. The point with us is that as Mr. Raghavan has told, we have designed various programs here, role model scientist discussion with role model scientists, visit to the labs, which is not possible due to COVID now. We would love if you can turn your camera towards your lab and then show them our equipment. Really, they will be really excited to see that. This is an inspiration part. As a matter of fact, every admission in the higher education depends upon the entrance examinations. So we also support them for taking the competitive examinations, call the expert teachers, and then make them prepare for the competitive examinations as well as. Although our students are performing very well, you will be very happy to know the average score of the students in Navodaya, as a matter of fact, for 70,000 children who went to board examination this time, 73,000 children, it is touching 80 average score. Yeah, so they're exceedingly performing well, but the expectation is that they should choose science as a career. Now, everybody wants the role model before them. So God-given opportunity, you are in front of them. Would like to bless them and show your lab and then share your research activities with the students. And I'm quite sure, ma'am, students will be very genuinely interested to know about the science you are doing. Thank you very much. Welcome, ma'am.
Thank you, sir. Thank you, sir.
You have to unmute your mic.
It's not possible to show the lab because we are not allowed to go into the lab right now because of this.
That is true.
So, because there is an age barrier that you can go into the lab. So I'll just speak to them.
Thank you. Thank you, madam.
So I'll share my screen and start. Well, I would like to thank all of you, Mr. Kannan, Mr. Srinivasan, Joint Commissioner, Mr. Kalavan, so for welcoming me. And it is indeed very exciting when you talk to young students and they pose the most difficult questions when they're out of school and they have read sufficiently, but not enough. So they have many queries. And so it is, so I'm quite thrilled to be here. And let us, I'll speak about, I chose a subject in a way that why studying science is an enjoyment. And I will partly give my story and also try to show how, how science goes on. So thank you very much. And I can now share my screen and start. Can you, is my screen visible?
Yeah, perfectly okay, ma'am. You have to run rather a slideshow, running slideshow.
Yeah, so this is okay?
Yeah, perfect.
Okay, so to all my young friends, when we talk about science, so that we want to inspire you to get into science, but let me tell you that some people do want to be poets, do want to be writers, do want to be painters, which is all right. But even if you are a writer, painter, or a poet, you should know a bit of science because science runs our lives. And science actually gives you an insight into the world, that how the world runs and what the world is about. So, so now let me tell you that what can science do? Science actually helps to solve the world's biggest problems. For example, feeding the world. By feeding the world, I mean that by 2050, we will have 9 billion people who the world will have to feed. So it is only science who through research and through application of basic research can find out how to feed the entire world through aggressive farming, intelligent farming, and that is the only way that we can provide nutrition to our population. And also we have to think about supplying energy, because our fossil fuels are running out slowly. So one day we will not have any fossil fuel, by which I mean oil, coal, etc. And so science will have to find a way, and we have made a significant progress on that way. Then where science also helps in curing diseases. So many diseases have been cured, so many cures have been found for diseases. But we also have to combat emerging diseases, as you can see now that we are combating the COVID-19, where the SARS-CoV-2 virus is causing havoc. And also science's major challenge is alleviating poverty. So that remains a challenge, because we have not been able to alleviate poverty from many parts of the world. So it is up to the next generation, to you all, to figure out how with intelligent planning we can alleviate poverty. And science certainly can improve our quality of life. And science finally influences our culture and our civilization. Now, this is not to say that what all happens in science is good, because there are collateral damage that happens with certain doing science in certain areas, and we have to find ways to, like, for example, we have polluted the atmosphere by inventing cars, by inventing, industrialization has happened. So we have to now, through science, find ways to mitigate these ill effects of science in that way. But science has given us the world, science has given us our life. And so science is a way of life, and we must look at it in a very measured way, so that we do not use science to spoil the world. We should use science to actually improve the world. So that is the message to you. Now, I just wanted to start with the coronavirus, because what has science given us? You might be thinking that science has not been able to tackle the coronavirus. But that is not true. If you look at it, that the first patient of COVID-19 was admitted to the Central Hospital in Wuhan on December 26. The sequence of the virus was available on January 5, so within a few days. Then several diagnostic tests were developed between January 20 and 30, and that is what enabled the governments to isolate people and prevent the spread of the disease so much. But we have to follow certain norms so that this disease doesn't spread. But actually, this enabling of able to prevent people from meeting each other came from the realization that this virus is very contagious, and that is a finding of science, and the sequence is a finding of science. Now, making vaccines is a finding of science. So that is how science has contributed in controlling this. And many more people would have died if we didn't use science to tackle this virus. Now, if you look at this, you see the virus actually attaches to something called ACE2 receptor on the surface of human cells. And this is what science understood. And this is a beautiful picture from a structural biologist who has shown that this is the spike protein, what you are seeing here. And this is the human ACE2 receptor. And all the residues have been figured out here, which is very important for designing an inhibitor, for designing a vaccine. So this is how beautifully that science has actually deciphered the identity of the virus and how it actually binds to the cells. So with that, you see that the coronavirus actually, as I said, binds to the ACE2 receptor. But that is not enough. There is somebody called TMPRSS2, another protein, which helps the virus to bind to this ACE2. This is like an insider who's doing a damage to oneself. And so it is pertinent to inhibit this protein to go ahead with suppression of the virus. And that can be done with inhibitors, and there are already inhibitors into trial. So you see how science very quickly, within 6-7 months of the infection, has come out with the places that where interference can be made through inhibitors to prevent the disease. So this is a contribution of science. And this is all very exciting stuff, irrespective of the tragedies that are happening with the COVID-19. But there is beautiful science that has been done. There are work which has contributed to significantly to alleviation of this disease. Now, should you become a scientist? So if you are a scientist, you are actually in a beautiful world where you ask questions, and when you find an answer, you are very happy. So I have spent my life doing science, and I find it very gratifying to come to the laboratory every morning to do something new. At the end of the day, find an answer and may not find an answer. So it may be a disappointment, it may be happiness that you can find. But it is that repeated questioning and repeated effort to find an answer is what is very exciting. And finally, you are able to serve society through your inventions and innovations, which is very important. You can serve society in many ways, but this is one important way that you can serve also. Now, coming to talk about myself a bit, because this is something that I wanted to show that how I became a scientist. So I was very much interested in the night sky when I was young. I used to look at the stars, and this is the Milky Way galaxy, and we are somewhere here, very tiny part of this galaxy. When we were young, the atmosphere was very clear, and we could see parts of the Milky Way, which you cannot see anymore. But if you go to the planetarium, you will see. And here, it is a great thrill to see something that is so far distant, mysterious, and we do not know about it. So it is very, very exciting. And I had a small telescope with me, which is not as sophisticated as this one, but I had a small one. I used to look at the constellation of stars. And it is at this point of time that I decided I want to become an astronomer. So I told my parents that I want to become an astronomer and nothing else in life. So one day my father he went to some store and he got me a small microscope. And when I saw the microscope, I then took pond water under the microscope, buccal mucosa into the microscope, and I saw a totally different world. And that is when I changed my mind, and I thought I will rather become a biologist than an astronomer. So this is how it changed for me. Astronomy still, interests me, and I regularly look at astronomical write-ups, popular write-ups, and I enjoy it very much, but I became a biologist in this way. Now, the biology is wonderful. If you look at the world around us, you will see more than 10,000 species of birds, more than 8,000 species of amphibians, mammals, more than 5,000 species, fishes, 30,000 species, reptiles, more than 6,000 species, and cylinderates, more than 1 lakh of species. Now, Charles Darwin, you must have heard of Charles Darwin. Charles Darwin said, "Endless forms most beautiful." So this is our beautiful world, but you must be aware that we are losing many species. There is a great loss of biodiversity because of our callous approach to our world, to the earth, and that is what we have to prevent. We have to really prevent this loss of biodiversity, and it is for you guys who are going to be the future scientists that you have to find ways to prevent the loss of biodiversity and preserve our world as it came to us. So this was the world that I entered into as a biologist. And I used to have a laboratory consisting of small bottles and collecting insects and plants all over the place. I used to do that when I was in school. And then I was enthralled by the the caterpillar turning into a pupa, and then the pupa is converting into a butterfly. That was one of the most intriguing things that I observed. So I sat with the caterpillar for a long time, and even at night, and they would at some point of time they would start to roll and form a pupa, and then eventually after a few days the butterfly will emerge. So this was another experiment of nature that really interested me and pulled me towards the biological sciences. Now, you see, when you look at biology, you also, when I was personally looking at biology and doing little experiments in school, I also was reading about the scientists. Not many books were available about Indian scientists or Indian women scientists even. So I read many biographies at that time, and those are called role models. So you have to go through many of these role models to look at what biographies and role model histories give you is they give you the problems that a person faced, how the person solved the problem, what did he or she do for a career, and finally it gives you a contemporary history of the period to which the scientist or even a writer belongs to. So I didn't have too many role models from the country at that point of time, but one person that I came to know was Dr. Janki Ammal. Dr. Janki Ammal was born in 1897. Actually, you can imagine 1897, about 125 years back. And she was born in a very conservative town in Kerala, Tellicherry. And so in Madras, and sorry, not in Kerala, in Chennai. And she grew up to study botany eventually. And she was one of the rare women who went to England to study, went to Michigan to do her doctoral studies. And she contributed a great deal to the genetics of plants. So she made, then she came back to India, and finally she made several intergenic generic hybrids to the sugar cane. And she actually made our sugar sweet for us, because before this, we were actually importing sugar. So you can imagine how much we were paying by importing sugar. So she actually made the sugar more sweet for us, and our imports actually went down. So finally, Pandit Jawaharlal Nehru called her to become to lead the Botanical Survey of India, and she was awarded the Padma Shri in 1977. And she was an out-and-out dedicated scientist. And even today, if you go to London and walk into Battlestone Hill Park, you will see the magnolias flowering in them, who were planted by Dr. Janki Ammal. And she died while working in the laboratory at the age of 87. So you can see such dedication, and she was a role model for me. There was another role model, which most of you have heard about. She is this lady, Marie Curie. Now, this is the family of Marie Curie. This is her husband, Pierre Curie. This is a daughter, Irene Julia Curie. And this is a son-in-law, Frederick Julia Curie. And now they have the highest number of Nobel Prizes amongst them. Both of them won Nobel Prizes. She won 2 Nobel Prizes. He won 1 Nobel Prize. So there was a very, very, extraordinary family, I would say. And what actually struck me about Marie Curie was that she discovered radium and polonium, the 2 radioactive elements. And she actually put radioactivity in use, because under her direction, the first experiments were conducted to treat tumors. And also, she was a very socially conscious scientist. And what she did during World War I was she developed a car, a mobile radiography van, I would say, and took it to the front, and it studied anatomy to do all the x-rays of the soldiers. So here is a scientist in the stature of Marie Curie who goes and works in the battlefield. So this was if this could not be an inspiration, then what could be? So there was this great Marie Curie. So these are the two women that actually inspired me to see the world in the light of science. So many years later, a few years back, I went to Paris and I went into Marie Curie's laboratory. And believe me, that it is kept just like as if Mary is going to step in. This is her, on the right-hand side, you are seeing her office, and on the left-hand side is a laboratory. And they have been preserved. Even Marie Curie's gown is preserved and a model that to show it to the public, and how beautifully it has been kept. So you see, so it was like a pilgrimage to me after I've dreamt about in the childhood how to be a Marie Curie, and then finally I go to her laboratory in Paris. So what I did was I did my MSc, where I studied cells in the brain, which was primarily a histological study, which I did at the University of Calcutta. Then I did a PhD on which how cells are killed in the reproductive tract was my problem. And I did that in Indian Institute of Chemical Biology in Calcutta. And then I did 2 postdocs, and for 2 years each. And one was how cell survival is controlled by hormones, and the other was how cells survive with small molecules, meaning the signaling pathways. And finally, when my dream came true, and I had my own laboratory, I started looking at how cells die. So what I did was, from Kolkata, I actually took a flight and went to the Middle East, where in the Kansas City Medical Center, I studied the signaling in cells. Finally, I came to New York City to do further studies on my postdoc. And then I came back to Delhi, not to Kolkata, but to Delhi, to establish my own laboratory. So as you know, we have cells in our body, and these cells, a group of cells, as I show you here, some cells are dividing, as you can see, some are in interphase. And amongst all these cells, not all cells may not be healthy. Some of these cells may be aging. Some of the cells may be actually sick through exposure to chemicals, exposure to carcinogens, to oxidative stress. So these are different cells of the body. You know very well, this is the red blood cells. These are the neurons that we have in the brain, which has a long axon. Then the liver cells and the kidney cells, and so the cells look very different, but they have the same DNA in the nucleus, except the RBCs, which are not nucleated. So this is a cell that all of you have studied, I'm sure, that this is the nucleus, the endoplasmic reticulum, the Golgi, and the mitochondria. So I'm sure you have studied it. And so even though the cell has been studied very extensively, there remains a lot of mystery about the cells. So I was interested at looking at how cells die. So I got interested in cell death, and I looked at cellular apoptosis. I do not know if you have heard about apoptosis, which means that cells are dying in a neat way. So when cells die, if they splatter and throw out their contents, what happens is that causes inflammation in the body. So apoptosis is a way by which cells are disposed of neatly, and there is no spillage of content. So here you see the cells are having small divisions which are membrane bound, and these are eaten up by the cells of the immune system, and when they eat up, there is no spillage. So apoptosis is a very neat form of cell death, and I wanted to ask several questions on apoptosis, but before I go into that, I just wanted to show you the actual hero of apoptosis. This is the nematode worm called C. elegans, and it's a transparent worm where you could label the cells and study the apoptotic pathway. So in 2002, 3 gentlemen, Sidney Brenner and others, got the Nobel Prize for deciphering the apoptotic pathway. So it's a very interesting thing that why cells would die and how cells die. Now, if you look at us, we have cells in the body which are normal cells, but then you have cells which are also dying. So our normal body consists of cells surviving and cells dying. Now, if all the cells were dying, we wouldn't have a body. But if cells, when new cells have to come and replenish these dying cells, and then we have a body. So there is this exquisite balance between dying cells, between living cells, which actually gives you your body. So how many cells do we have in your body? We have about 100 trillion cells in our body, 14 zeros, 100 trillion. And we have about 1 billion new cells per day. So if there are 1 billion new cells per day, there must be equivalent number of cells which are dying to give space to these new cells. So this is a never-ending procedure that keeps on happening in all animal bodies. Now, as I said, that there are many cells that die in an adult human per day. And in a given year, the mass of cells that we lose from our body is almost equivalent to our entire body weight. So you see, we are a new us at the end of the year because we have lost so many cells. The cells are either aging or are hurt by some insult, so they have to die and the new cells have to be replenished. So now this is the photograph of a dividing cell. It's a scanning micrograph, and this is the photograph of a dying cell. So a dying cells, as I said, they form these blebs or round like membrane bound forms which are engulfed by the immune cells and cleanly disposed of. So these are two very grand pictures of a normal cell and a dying cell. So why is this cell death important to us? Now we arrive as a single cell. When the sperm fertilizes the egg, we get an embryo. So this is a 2-cell embryo. Actually, it starts with a single cell embryo. And so this is how we all start. I mean, all animals start as a single cell. Now, this single cell then divides into a ball of cells, which is known as the morula. So this, at one point of our lives, we are a ball of cells. Now, this ball of cells, if it kept on growing as a ball of cells like this, what happens? We will all be a ball of cells without hands and feet and face and all that. So what has happened is the cells now keep on dying. So when the cells die, they give you this shape. Without cell death, we cannot give this shape, cannot get this shape, and abnormalities of this cause problems. So if you look at a stone, piece of stone, when a sculptor is chiseling out at this piece of stone, from this kind of piece of stone, he will or she will get a sculpture. So it is just like that for our body, that the cells will die and sculpt the whole embryo. So this is why during development, it is very important that cells do die, and some cells also keep on dividing and coming at that time. And if you look at the tadpole, the life of a frog, the tadpoles, you see the embryos, the tadpoles, you must have read about the tadpoles. The tadpoles have tails, they have to swim in water. So eventually what happens is they lose the tail. And this is by apoptosis, that this tail keeps shrinking. Eventually the frog is born without a tail. So this is also cellular apoptosis, by which an important event takes place, the loss of tail by the frog when it comes from the water to the land. It can still be in the water, but where the tadpole cannot survive on the land. It survives only in the water. So if you look at this, these are the apoptotic cells that are happening in the tadpole, and eventually the frog becomes like this. Now we are born with our hands like this. So there are actually inside our fingers, there is actually a web, and the cells of this web have to die to give us the shape of a hand. So look at this, that these are the cells that are undergoing apoptosis. And what happens if they don't die? You get joint fingers, It may be in the hand, it may be in the feet, but this is only the exterior part of our body. But when you actually look at the when you actually look at the internal organs, when there is a problem with cell death, then you get tumors. So so it is very important that we get a proper cell death. So you see here, this is a chicken leg and this is the duck leg. So the duck has to swim, so cells don't apoptose here. But the chicken has the leg which cells have died, and it gives it the leg it needs to walk on the land. So this is how cell death is very important. And if you look at consequences of anomalous cell death, you will see that you get neurodegenerative diseases. You have heard about AIDS, where T helper cells actually die excessively, and so you can get AIDS. AIDS actually is because of apoptosis of the T cells of the body. And then you can get infertility when germ cells would die excessively. So these are excessive cell death causing these problems. But if you have a diminished cell death, then you can get tumors and you can get autoimmunity, and you can also have neurodegenerative diseases. So the point that you have to look into is that if there is excessive cell death, there is a problem. If there is diminished cell death, there is a problem. So it is very important that there is a very critical balance between the excessive cell death and diminished cell death. So if you look at this set of cells, you see that there are cells that get mutated during our life. And when they get mutated, what happens to a cell which has accumulated mutations? So it has become a cell which is not normal. So under the circumstances, the body tries to get rid of that cell. So if the body actually fails to get rid of that cell, what happens that the mutated cell keeps multiplying, and once it multiplies, it becomes a tumor. So you get a tumor. So it is important that your body gets rid of that mutated cells. That doesn't happen many times, and you end up getting a tumor. So these are some pictures of the tumors. And in the body, the tumors can also be killed by apoptosis by our immune cells. So these are T cells, cytotoxic T cells, that are actually attacking a tumor. And when the tumor has to shrink, cells have to die in the tumor by our body's own effect. So in my laboratory, I asked several questions, that when did cell death evolve, and how is cell death important for us, and can we manipulate cell death to treat diseases, and then do single cells die by apoptosis? So if I can confine myself here, that is a very interesting problem that we addressed, that if you come to think of it, why should a single cell die? See, single cell we see all around us, and even the bacterium is a single cell. So if a single cell die, it kills itself. So it goes against the principle of evolution. So because evolution helps us to live. So what is it? Why does it die? So that question, we do not know the full answer. But what could have happened is that when single cells get together, they attain a state of pseudo multicellularity, and that's when they can afford to have one cell dying. So we did many experiments with it, and we got into a disease which is called leishmaniasis, which is a part of a neglected tropical disease, and it affects 1 billion people across the world. And this is how the parasite looks like. It has two forms. It has a elongated form that is found in the humans, and this is that lives in the gut of the sandfly, and is also found in the humans when the sandfly infects the body. And then you get the rounded forms when they are inside the mammalian body. So this is the infection. This is the cell that they are infecting, and when they go to infect, there are many in numbers, so that the infection becomes successful. And it happens when the sandfly bites our skin. This is the skin. This is the layers of the skin. And the parasite gets in and tries to invade the body. Eventually, the body tries to kill it by generating an oxidative burst, which you may have read, which is generated by reactive oxygen species inside the host cells. And they try to kill the pathogen, whether it is a parasite, whether it's a bacterium, whether it's a virus. And when it is successful in killing the pathogen, there is no disease. But when it is unsuccessful in killing the pathogen, then there is disease. Now the pathogen tries to, I'll come to that in a bit. Here the pathogen actually is inside something called a phagolysosome in the body. And this phagolysosome generates a lot of reactive oxygen species from oxygen, from reactive oxygen. And it actually affects the cell membrane, it actually affects the nucleus, the internal organs, and eventually kills the pathogen. Now it depends, when you get a disease, that means you have not been able to kill the pathogen. But when you are able to kill the pathogen, you are free. So it is very difficult to find out how much you have been able to get rid of and how much you have not been able to get rid of. So when you get sick, you know that you have not been able to fight the pathogen. Now, if you look at this picture I'll show you some pictures that we generated during our, the details of the work is where you will probably not be able to enjoy. But these are the little phagolysosomes. And stained red means they are lysosomes, and they are stained green means they are producing the reactive oxygen species. These are with special dyes, and two together means that you are seeing exactly at that point of time where reactive oxygen species is being generated in the phagolysosomes. So next, what we did was, again, in the phagolysosome, the parasite survives like this, and you see exactly that we have produced here a stained version of a macrophage, that is an immune cell of the body with the parasites. And then they are getting killed by oxidative stress, which are stained with special dye, which shows that the parasites are dying. They are again single cells which are dying. And this is the photograph of a Leishmania parasite, where this is the mitochondria. These are the isolated mitochondria, where we have shown that the mitochondria is very important for apoptosis in Leishmania. Mitochondria, is the powerhouse of the cell, but it has other functions also. And so if the mitochondria actually regulates whether the cell will survive or the cell will die by apoptosis, because they can release many factors that leads the cell to apoptosis. So why it happens? So survival within a host cell, what we studied was when any pathogen invades a host cell, the first impulse of the host cell is to commit suicide. That means they kill themselves. So that when they kill themselves, they're eaten up by the body's immune cells. And when it happens, the cells are disposed of and the pathogen cannot survive. But when there is a failure of host cell's ability to kill the pathogen, then the host cell actually dies by itself. So, so there are two ways. One, it succeeds in killing the pathogen, or one, when it doesn't succeed, it actually dies. Then another very smart way that these pathogens have acquired is that the whole signaling pathway of apoptosis can prevent that. So if they can prevent that, they can prevent this part, that they will not let the cell die. The pathogens are so smart that they won't let the cells die and the cell will survive. So it is this tussle between the pathogen who wants the host cell to survive till it needs to go out, and the host cell's desire to kill the pathogen. So natural selection has worked on both, and that's why the pathway of this cell survival and cell death has evolved. So now, coming to science is a very enjoyable activity. You saw the beautiful data we have produced, and I've shown you very little bit of it. But we have more data, and if you read the papers, you won't be able to understand right now. But in a popular science form, you can perhaps understand the host pathogen interaction. Now you have an idea, you go to do research, finally you get your data together and look at it. So it's the whole day, it goes like this, the whole week goes like this, the whole month can go like this. It can take years for you to make a discovery, but it is all great fun because you are doing something very exciting. Now, it is a bit like cricket. So in cricket, what do you need? You need discipline, endurance, strength, skills, and patience. So there are a set of rules. So all these comes in science, that you need discipline in science for science to work for you. You need endurance because you have to have long hours in the laboratory. Very exciting. Then you have to have strength. Now you have to have strength to survive that struggle. And then you have to have skills. That skill you have to acquire in the laboratory, and you have to have great patience that it requires. But all these are virtues of life that we enjoy. So now, see, there are moments of success and disappointment. You look at this guy who's caught a ball and has a successful catch. He is successful, so there's a moment of success. Now this is when the catch is dropped, and this is by Rahul Dravid, one of the most successful catchers. He has dropped a catch. So that's a moment of disappointment. And here you see collaboration. Collaboration is another thing in science. Here all the fielders, the wicketkeeper, and the bowler, who you cannot see, actually is teaming up to do some collaborative effort. So teamwork triggers creativity, and people tend to share ideas to be more open. Teamwork triggers learning. Teamwork helps develop bonds that may last long. So that is why you should get into a team sport. Team sport is very important. This is a very old picture of mine. Here I am in this. I used to play cricket, and I represented my state for 3-4 years while I was studying. I was doing this also. So it is a great to work in a team sport, so that in the laboratory also you have a team going for you. So these are all the members of my laboratory who have gone through in various years. So I've been able to work with this great team of workers. Now, I wanted to show you one role model. There's a modern day role model, is Dr. Sarah Gilbert. She's one of the scientists that is leading the race to find a coronavirus vaccine. And she's a vaccinologist, and she's at the University of Oxford. But she has triplets, meaning that she has 3 children, and she had to bring up those children while doing the science. And she has injected all the 3 children with the test vaccine. So you see how a modern day role model can be read about and get inspired for. So she is a modern time person, but she has managed her family, she has managed her science. So it is entirely possible to manage your family and the science and do such great service for the society that you see. Now, how far have we come with science? Now, science has a massive impact on our lives. In the last 50 years, there have been many discoveries. One of the most foremost discoveries has been the understanding of the DNA structure. And this has really helped building knowledge about genetic diseases, about forensic uses, about identifying infectious agent. As I told you, that within a few days, the coronavirus genome was sequenced, and we could, understand the virus. Then there are little things like your ATM machine, the barcodes, the fiber optics, the robotic surgery, the MRI, which actually gives you how modern technology has evolved and made our life different. But let me tell you one thing, that behind all these innovations, there is very, very strong basic science. Without basic science, nothing works. So one has to work on basic science in a way that you get into a problem, you make a very good fundamental basis for you to build upon, to do the innovation. That innovation you may do, that somebody else may do, but it is extremely important that you have a very strong basic science. And basic science is great fun. Innovation is also a great achievement. Finally, application to the society is also important. So I think this is how I thought I would tell you that how science goes about and how it can be very exciting. And so I would stop here and I would take questions. Like solar energy is a new thing that has, new thing in the sense that it's been around, but it has improved more and more. The GPS that we use is something that was not there, a few years ago. This made our life very easy. Then biofuels have come so that we do not pollute the atmosphere. Then mobile phones, when we were young, we did not have television, we did not have the mobile phones, but we lived, we read books, we, talked amongst ourselves. So the times have changed, but science has helped us with innovation. Science has also helped us to build our world, but at the same time, we have to see how we also take care of our world. Thank you very much. I can take questions now.
Please, madam.
Yes. Do I have questions or?
Madam, children are getting ready, madam. In between, one small doubt for me, madam.
Yes.
Somewhere else in the report they are mentioning this coronavirus is not at all a virus, it is a bacteria. Any idea, madam?
No, that is not true. Actually, it has been established as a virus because we make, we sequence the genome and we know whether it's a virus or a bacteria. So it's a bacteria is not true, actually.
Not true. Okay. Thank you, madam. Thank you. Thank you. Nagabhaskar, sir?
Yes, sir. Yes, sir. Good afternoon, madam.
Good afternoon.
Yeah, madam. Actually, we know that the climatic change lead to the extinction of many species.
Yes.
Yeah. Does this climatic change do have any effect on the rate of mutation that lead to the evolution of a new type of these viruses? Any study has been conducted, madam?
You see, climate change, in a sense, also generates many indirect effects.
Yes.
Indirect effect on our cells, indirect effects in the germ cells, and that do produce mutations, and that can lead to emergence of new species. And that doesn't happen, that happens over billions of years. So along the evolution that what we have seen for many millions of years over the time scale, evolution did to evolution, some of the climatic factors have contributed, certainly.
Madam, because these are the RNA viruses, just now which emerged as instead of COVID, because they are more subjected to the change. RNAs are more subjected to the changes. I mean, mutation rate of the mutation is faster compared to the other viruses.
Yes, coronavirus mutates slower than other viruses somehow, because they do not mutate very rapidly. So it may be easier to find a vaccine, because mainly why you can't find vaccines for the viruses is because they keep on mutating very fast. But this degree of mutation also depends on different kinds of viruses, and coronavirus apparently do not mutate as fast as some viruses.
But within it, from December to this, as per our knowledge, there are something 9 to 10 subtypes have been created, evolved in the coronavirus.
9 to 10?
9 to some subtypes.
Oh, subtypes, yes,
Yeah.
there are subtypes.
Okay.
Because you see, a virus, it mutates, and I'm not saying coronavirus does not mutate, it mutates. So it gives rise to subtypes or clades, as they call it. And then you can find these subtypes. And it has happened with this SARS-CoV first virus, then MERS, that they have evolved into less virulent subtypes. It has happened.
Madam, one more small clarification I was doubt for me, because you mentioned this cell death, excess of cell death lead to neurodegenerative disease, that is understood. But how, madam, this diminished cell death also lead to the,
Yeah, diminished cell death is what I meant was that in the brain, when we are born, in the brain we are born with many cells that we don't need. extra cells that we don't actually need for laying down the proper circuitry of the brain. And that is where cell death is required. So if the cells don't die properly and when our circuitry is being laid, then we do get anomalous, brain function. So even if there are less cell death, there will be there will be abnormalities in brain. And if there are less cell death, there will be also abnormalities in the brain, and which is primarily during development.
Thank you, madam. And this is what can be a triggering for this program cell death, apoptosis, in addition to this? What is it? It has a natural phenomenon. It is an inbuilt in a human, because if we speak about the RBC, it is having lifespan. Every cell do have a lifespan.
Every cell has a lifespan, and at the end of the lifespan, when they're aging, they will die by apoptosis. And otherwise, during development, I told you that the cells are programmed to die to give shape to the body. And that is one. Otherwise, cells can die by our exposure to the UV radiation, exposure to pollutants, exposure to insecticides. They also kill cells. And they kill cells, they also mutate cells, so that you will get tumors or cancer or so much cancer because of our environment. Many the environment gets polluted, and there are inherent, some of the cancers are hereditary, so you do get hereditary cancers also. So it can be inflicted upon you, or you can have inherent built-in program for your cells to turn cancerous.
Last question, because these viruses are also an intracellular parasite. Okay, generally the parasites are very specific to the host cell, but we have seen these viruses. They're zoonotic. From an animal, it has been started entering into the human transmission.
Yes.
How they, what things they change it? Because even that host is also available, because pangolins are also available, bats are also available. But why? What makes these viruses to move to get an alternate host in addition to this pangolin as well as the bats and coming into the human?
Well, this is a process of natural selection, because as the viruses grow, they try to find new hosts because they want to survive. They want to do whatever they want to do. So they went to the pangolin. The pangolins were infected. We do not know if other animals have been infected. So from the pangolin, they came to the bats, and the bats gave it to the humans. Now that is also true for example, the HIV virus. It was in monkeys. It came to the humans. So they are always trying to find a new host, and the mutations do change and make their host specificity change. So that is how the species jump actually. So always it has been attributed to some eating of the meat, or even in HIV, eating of drinking of monkey blood or something. Some stories are there, but it actually jumps species in some kind of contiguous form. It comes to the body, to another body, and make its home in that new body. So exactly when and where it happens is very difficult to find.
Thank you. Thanks a lot.
Hello.
Yes.
Ma'am, I'm having a doubt.
Yes.
We are hearing from different countries that they have made a vaccine for COVID.
Yes.
We also hear that the COVID virus also is mutating.
Yes.
How is the research? How are they accommodating everything to changing situations? And different countries are attempting at their own levels. Will all the vaccines contain the same ingredients, or will it be totally different?
No, these are different ingredients. Like the vaccine that now Oxford is giving, it is an adenovirus vector, which is a chimpanzee adenovirus vector, which has the spike protein of the coronavirus inside it. So because the spike protein binding is very important, many of the vaccines are using spike protein, but either in the adenovirus vector or in another kind of vector, or RNA vaccine. Now, RNA DNA vaccines has not been approved so far, but RNA DNA vaccines that people are also using. So there are different kinds of vaccines that are coming up. Now, one of the vaccines that China has tried is just heat killed, I think, heat killing or killed virus vaccine, where the whole virus is there, but it's killed and it is injected. Like if you remember that there was smallpox vaccine, and the smallpox vaccine we used to have, we used to take, I don't know, I am much older than you guys, so we used to take a vaccine every year, and these were all killed cowpox viruses. So this was a very, very successful vaccine. So in this way, China has actually injected, I think, some people in the army with this vaccine. The whole vaccine, the whole virus killed and used. But there are adenovirus vectors they are using. So there are different kinds of vaccines that are coming up. Which one is going to be successful, we don't know. But
Thank you.
the best bet seems to be the killed virus, although it's quite dangerous to do that. But
Okay. Thank you, ma'am.
Good afternoon, ma'am.
yes.
My name is Trisha, completed my 12th this year. I have a question for you, ma'am.
Yes.
So, why do cells change the way they do? Like, for example, how normal cells change to tumor cells and what makes it happen?
Okay. So normal cells, as I said, they accumulate mutations, right? So all our cells sort of accumulate mutations, and sometimes they are irrelevant mutations, sometimes they're relevant mutations. So, or the cells also get bombarded by UV or other agents. So cells, there is a cell death process. If the cell is sick, the cell death process will kill the cell. But as it happens with pathogens, that they prevent the cells from getting killed by interfering with the cell death pathway. So similar things happens in the cancer cells, that a cell all of a sudden loses its ability to initiate apoptosis, to initiate cell death. So it survives. And when it survives, it grows rapidly, because that's the characteristic of the rapidly dividing cancer cells. And so that becomes a tumor. So you actually basically lose your ability to initiate a cell death. And that is why you keep on dividing and generating these tumors.
One more small doubt, ma'am. Like, in case of amoeba, the cells divide and we say they are immortal cells. So the same way even somatic cells divide, no ma'am. So why are we not considering it as immortal? And does these have any condition where they stop dividing and die?
Yeah, our cells in the body go to about 50, 60 cycles of cell division. It cannot go not more than that. So they are actually very mortal, and they die at some point of time. That is why the replenishment of cells takes place here. But amoeba, being amoeba, it's a clonal division. So there are many, unicellular organisms that go through clonal division. But every time they divide, they have some little bit of change. But we cannot call ourselves immortal because of that they have a defined number of cycles they can divide. Beyond that, they cannot divide. But there was one instance of this HeLa cells. Have you heard of HeLa cells? I don't know. These
No.
HeLa cells came from a lady who had a cervical cancer, and her name was Henrietta Lacks. And these cells kept on dividing, and it actually was distributed to various laboratories in the world. And that cell, after she died in 1952, I think, and the cells are still alive. So that is a super immortal cell. So many cancer cell lines have been generated to by transforming them and having the immortal lines. But Henrietta Lacks cells were like immortal by themselves.
Thank you, ma'am.
You're welcome.
Madam, I believe Professor Abha Misra is on board with us. Madam, would you like to share anything, madam, with us?
Where is she? She's with you?
Yes, Professor Abha Misra, madam.
She's there. Okay.
She's
Well, I
there.
would like to thank her for writing to me and giving this opportunity to speak to the children.
Yes, madam. Now, madam.
Miss, I really appreciate your time, and this is a wonderful lecture you have given, and the main motive of this lecture is for these girls who should be, Miss, who of course, Miss, why there should be kind of, so Miss, you are the main inspiration and the role model in our country. So I'm really grateful to you spending time with these children. And what I believe is there are 3,000, 4,000 students currently are watching your lecture, but suddenly maybe we could have more effective program for the direct interaction. Maybe in future we'll extend this program, and you may be able to interact with these girls directly and really give this motivation.
Yes, I think, they will understand that science is a lot of fun, and that with the fact that women can also do science even if they have a family. That's why I showed the example of Sarah Gilbert, because she's so famous and she's producing this vaccine and useful thing for society, and she has children she has brought up. And so a support system you of course need for doing this, but then it is possible to do. I mean, that's what I wanted to emphasize, that do good science.
So this is very important that Miss, people like you should actually tell these things, and this is like a direct message to them that look, nothing is difficult, and they really should not be worrying about anything for their future and deciding their career. So that is the motive of this program which DST has come up, and certainly with these lecture, Miss, today's lecture, it is really a wonderful lecture what you have given. And I'm hoping that so one more thing I wanted to Miss, I may also write you later that to have more interaction with these girls, maybe we can take their questions and we can have some kind of interactive session based on their questions and their worries, so that we get to know that what they think, what are their constraints, and
yes, see, sometimes we fail to talk to these people, these children, and we don't know what their viewpoint is. So if we come to know about their viewpoints, all the stakeholders, what we call quote unquote stakeholders, if we speak to them, we would know what they actually want, to know. Many things that their parents or teachers may not answer.
Truly. So we thought of extending this series to that level also, where means they should get chance to speak to you directly.
Yes, it can be easily held in a Zoom meeting. Actually, in a Zoom meeting, one can do that. Not many, but maybe 50 people.
Yeah, limited number. Yeah,
yes.
yeah. Actually, I also take the opportunity to thank our olive branch ma'am Abha Mishra ji. She has been helping us in coordinating all this, madam. Thank you very much. Madam, actually, our commissioner was also with us. He wanted to personally thank you. Although we are sitting in Delhi altogether, you know the difficulties. Sir has just left for a meeting with the secretary, so he couldn't join us. Thank you both of you for kindly helping our students, and I feel that this will go a long way in inspiring our student community. This will definitely ignite their power to involve themselves and then take the science study more seriously.
Yes, that's why I actually told them to read about scientists. And nowadays people don't read, they listen to YouTube or something. It's fine. I mean, there are good, videos on scientists' life, which they can actually listen to, and they will know. And of course, role models, as you're saying, I think if I could have inspired even one person, it is very nice.
More than enough. we are happy even if one person comes out of this. Yeah.
Okay, sir. Madam, just I would like to request my friend, colleague, and the co-organizer, Mr. Srinivasan, principal from JNU Mangalore, to propose a formal vote of thanks to our guest. Okay, madam? Okay. Srinivasan, sir, over to you, please.
Yes. Good evening, everybody. At the outset, I thank Professor Chandrima Shaha for having given us a wonderful, exciting, and enriching lecture. She has touched upon all that is required for the children to learn science in the way it should be learned. How she was having a passion right from her childhood to learn the macroscopic bodies using a telescope, then shifted her focus to the small is beautiful, the smaller ones through the microscope, and how she was taking the lead from the life of Dr. Janaki Ammal. And she also has touched upon the various ingredients for a quality learning in science, connecting it with the famous sport of India, cricket, how discipline, endurance, strength, skills, and patience, and also, very importantly, how to handle both success and disappointment. And she also insisted how that collaborative effort, collaborative learning is important for science learning. She also has very aptly touched upon the idea that even women can do wonders by citing the work of Dr. Sarah Gilbert. Ma'am, it was really a wonderful, thought-provoking session. I think our girls are very fortunate to have listened to you. So thank you for giving us your quality time. Thank you so much, ma'am.
Thank you. Thank you.
I also thank Professor Abha Mishra, Department of Applied Physics, IISc Bangalore, who happens to be the nodal officer, for having arranged such a wonderful talk by a wonderful person who is heading the Indian Science Academy. Thank you, ma'am. I also thank the Navodaya Vidyalaya Samiti and the Department of Science and Technology for having extended this wonderful Vigyan Jyoti project to our Navodaya girls. I also thank our Honorable Joint Commissioner, Dr. Ramachandra. He is always a guiding spirit, and he always encourages us to have such kind of program. So I thank him also for having participated in today's lecture. And I thank all the children, those who have given a good audience. I hope you must have taken the cues from what ma'am has told. It was an information-packed 1 hour. It was lovely listening to this wonderful woman. Thank you, ma'am, for giving us a wonderful session. Thank you so much.
Okay.
Thank you.
Thank you, sir. Thank you. Madam, anything more, madam? That's we can.
Thank you all. Thank you all very much.
Thank you.
I enjoyed it. So
thank you.
yeah,
Thanks a lot. Thanks a lot.
Sometime next.
Please. It is our honor to have you. Sir, anything, sir? Nothing. Okay. Thank you, madam. It comes to an end. Okay. Thanks a lot. Please.